Continuation of the previous video about immunology. So here we could see a picture which depicts a simplified depiction of the human MHC showing regions and genes coding for polymorphic MHC class 1 and 2 molecules, beta 2M encoded outside the MHC, adapted and modified from immunology A. Then a set of MHC class 1 plus class 2 genes present on an individual chromosome referred to as haplotype is passed on as a unit into the offspring. The offspring will also inherit a set of MHC class 1 plus class 2 genes for their other parent. Each individual expresses a distinct array of MHC class 1 and class 2 molecule. This diversity comes about because different individuals within a species have a range of slightly different forms or alleles of MHC class 1 and 2 class 2 genes, genetic polymorphism. Because of the extensive polymorphism of MHC genes, every individual has an at most unique array of inherited MHC genes. So within one individual, the MHC class 1 and 2 molecules expressed are the same on all cells of the body, and they are codominantly expressed at the cell surface, products of both maternal and paternal chromosomes. So here we could see a table which represents features of class 1 and class 2 MHC molecules. Class 1, structure alpha chain and beta 2M, polymorphic domain. Structure in case of class 1 MHC, alpha chain and beta 2M. Class 2 MHC, alpha and beta chain. Then polymorphic domain, alpha 1 and alpha 2 domains, alpha 1 and beta 2 domains, binding site for T core receptor, alpha 3 region binds CD8, B2 region binds CD4. Then constitutive cellular expression, nearly all nucleated cells, APCs, and peptides derived from endogenous antigens, exogenous antigens. Peptide presented to CD8 plus T cells, CD4 plus T cells. Then nomenclature, here we could see. Then antigen processing and presentation. Pathogens such as bacteria and viruses can penetrate and infect the cells, and T cells mount an immune response against the cells harboring the invading organisms. How do T cells recognize an infected cell or indeed the presence of any foreign protein antigen that is inside a host cell? The T cell receptor on T cell and antibody on B cells are somewhat related with minor differences. The difference between T cell receptor and antibody is that T cell receptor does not recognize free antigens. So T cell receptor recognizes only antigen that is bound to cell membrane protein called MHC molecule, termed as antigen presentation. So antigen recognition by B and T cells. Antigen interaction, B cell receptor membrane Ig binds Ag. Then T cell receptor binds Ag and MHC. Nature of antigens, protein, polysaccharide, glycolipid, and peptide. Then binding soluble antigens. In case of B cells, yes, but in case of T cells, no. Then epitopes recognized accessible areas of protein structure containing sequential amino acids and non-sequential amino acids in case of T cells. But antigens processed internally and presented as linear peptides bound to MHC molecules in case of T cells. And peptides of antigens associate inside the cell with MHC class 1 or class 2 molecules, and the peptide MHC complex moves to the surface of the cell where it can be recognized by a T cell with an appropriate receptor. In the presence of co-stimulatory signals, the T cell is activated and molds a response against the antigen. Events involved in the generation of peptides from proteins inside cells, the binding of peptides to MHC molecules, and the display of peptide MHC complexes at the cell surface for T cell recognition are known collectively as antigen processing and presentation. MHC molecules play a central role in these phenomena, and for this reason, T cell responses are said to be MHC restricted. In every vertebrate species studied, the function of MHC class 1 molecule is to present peptides derived from protein antigen to T cells. By contrast, the function of MHC class 2 molecules is to present peptides to Th cells. Then cytotoxic cell, T cell receptor, class 1 MHC protein, fragment of protein foreign protein, antigen presenting cell or target cell, helper cell, class 2 MHC protein, fragment of foreign protein. The recognition by T cells of foreign peptides bound to MHC proteins. Cytotoxic T cell recognize foreign peptides in association with class 1 MHC proteins, whereas helper T cells recognize foreign peptides in association with the class 2 MHC proteins. In both cases, the peptides MHC complexes are recognized on the surface of an antigen presenting cell or a target cell. So by convention, cells that display peptides associated with class 1 MHC molecule to CD8 plus TC cells is referred to as target cells, and cells that display peptides associated with class 2 MHC to CD4 plus TH cells are called antigen presenting cells, but sometimes we use APC for both. So how do peptides derive from proteins antigens associated with MHC molecule? Protein antigens presented by APC belong to two categories, exogenous and endogenous antigens. So endogenous and exogenous protein antigens use different pathways for processing and presentation to cytotoxic T cells and helper T cells respectively. So human red blood cells are not nucleated and do not express any MHC molecule. Why is this property fortuitous for blood transfusion? For RBC expressed MHC molecules, an extensive tissue typing would be required before a blood transfusion, and only a few individuals would be acceptable donors for a given individual. So explain the differences between the terms antigen presenting cell and target cell as well as they are commonly used in immunology. So by convention, antigen presenting cells are defined as those cells that can display antigenic peptides associated with class 2 MHC molecules and can deliver a co-stimulatory signal to CD4 plus TH cells. Target cells display peptides associated with class 1 MHC molecules to CD84 TC cells. Then cytosolic pathway. Endogenous antigens are synthesized inside a cell. Typically, they are derived from pathogens, viruses, bacteria, and parasites, and have infected the cell. MHC class MHC proteins present peptide antigens derived from endogenous protein antigen. Processing of endogenous antigens occur in the cytoplasm rather than in the acid vesicles. The major mechanisms for generating peptide fragments in the cytoplasm is via a giant protein complex known as proteasome. This cleaves proteins into peptides about 15 amino acids in length. So cytosolic enzymes, aminopeptidases, remove even more amino acids from the peptides. Peptide antigens are transported by an energy-dependent reaction into the cytoplasmic reticulum through a pore formed by proteins called the transporters associated with the antigen processing tap. Once the peptides have entered the ER, they are bound by the MHC1 protein held in the place near the TAP site by a group of chaperone proteins. The MHC1 peptide complex is then released from the chaperone and moved to the cell surface where it integrates into the membrane and can be recognized by TC cells. So endogenous antigens plus ubiquitin ATP cytoplasmic proteasome complex, then peptides TAPER peptide class 1 MHC complex. You could see a picture which depicts this. The processing of an endogenous protein antigen for presentation to a cytotoxic T cell. Class 1 MHC proteasome endogenous antigen. Endocytic pathway. Like the endogenous antigen presented to cytotoxic T cells, the proteins presented to helper T cells on APCs are degraded fragments of foreign proteins. Rather than being derived from foreign protein synthesized in the cytosol of a cell, the foreign peptide presented to helper T cells are derived from endosomes. These peptides come from extracellular microbes or their products that the APCs has endocytosed and degraded in the acidic environment of its endosome. So processed antigens not enter the lumen of the ER where the class 2 MHC proteins are synthesized and assembled, but instead bind to pre-assembled class 2 heterodimers in a special endosomal compartment. Once the peptide has bound, the class 2 MHC protein alters its conformation, trapping the peptides in the binding groove for presentation at the cell surface to helper T cells. A newly synthesized class 2 MHC protein must avoid logging its binding groove permanently in the ER lumen with peptide derived from endogenously synthesized proteins. A special polypeptide called the invariant gene ensures this by associating with newly synthesized class 2 MHC in the ER. Part of its polypeptide gene lies within the peptide binding groove of an MHC protein, thereby blocking the groove from binding other peptides in the lumen for the ER. The invariant chain is cleaved by proteases in the late endosome. However, a short fragment of an invariant chain termed clip for class 2 associated invariant chain peptide remain bound to the class 2 MHC, preventing any premature binding of antigenic peptide. This fragment is then released, catalyzed by a class 2 MHC-like protein called HLA-DM, freeing the MHC protein to bind peptides derived from endocytosed proteins. Exogenous antigens, exocytosis or phagocytosis, endocytic compartments, peptides, peptide class 2 MHC complex. Invariant gene, exogenous antigen, the processing of an exogenous protein antigen for presentation to a helper T cell. Next major topic is about immunoglobulins, structure and function. Antibodies, the antigen binding glycoproteins are synthesized exclusively by B cells and in billions of forms, each with different amino acid sequence and a different antigen binding site. Collectively called immunoglobulins or IG, they are among the most abundant protein components in the blood, constituting about 20% of the total protein components in the blood plasma. Antibodies are present on the B cell membrane and also are secreted by plasma cells. So, basic structure of antibody molecule. The simplest antibodies are Y-shaped molecules with two identical antigen binding sites and at the tip of each arm of the Y. Because of the two antigen binding site, they are described as bivalent. Antibody has a common structure of 4 polypeptide chains. It is a heterodimer consisting of two light chains, each containing about 220 amino acid residues, about 25,000 mW, and two identical heavy chains, each usually containing about 440 amino acid residues, about 50,000 megavolt. Each light chain is bound to heavy chain by disulfide bridges and other non-covalent linkages. This an antibody is a dimer of H2L chain. All species studied have two major classes of light chains, and any one individual of a species produce both types of light chain. However, any one immunoglobulin molecule, the light chain are always either both K or both lambda, never one for each. Well, there are two types of light chains. The immunoglobulins of virtually all species have been shown to consist of 5 different types of heavy chains, alpha, beta, like that. Okay, these 5 different types of heavy chains are called isotypes. The heavy chains of a given antibody molecule determine the class of that antibody, IgM, mu, IgG, IgA, IgD or IgE. Each class can have either K or lambda light chains. Any individual of a species makes all heavy chains, but in any one antibody molecule, both heavy chains are identical. Thus, an antibody molecule of the IgG class could have the structure with two identical K chains, kappa I guess, and two identical gamma heavy chains. Alternatively, it could have a structure of like this one. So, both light and heavy chains have a variable sequence at their N-terminal ends, but at a constant sequence at their C-terminal ends. Light chains have a constant region about 110 amino acids long and a variable region of the same size. The variable region of the heavy chain at their N-terminus is also about 110 amino acids long, but the heavy chain constant region is about 3 to 4 times longer, or depending on the class. It is the N-terminal ends of the light and heavy chains that come together to form the antigen binding site. The diversity in the variable regions of both light and heavy chains is for the most part restricted to 3 small hypervariable regions in each chain called complementarily determining regions or CDR. The remaining parts of the variable region, known as framework regions, are relatively constant. Only the 5-10 amino acids in each hypervariable region form an antigen binding site. So, reduction of AB structure, Porter and Edelman elucidated the basic structure of Ig molecule, but their approaches were different. Here we could see a picture which shows disulfide bonds are shown, L chain, H chain, pepsin digestion will be taking place, FC fragments, pepsin, papain digestion, then mercaptoethanol reduction. So, prototype structure of IgG showing chain structure and inter-chain disulfide bonds. The fragments produced by various treatments are also indicated. Light chains are in grey, heavy chains in black. So, Porter found that proteolytic treatment with the enzyme papain split the Ig molecular weight given into 3 fragments of about equal size. 2 of these fragments were found of equal size. Each of MW 45,000 is called Fab fragments. What was that? Fab fragments. Okay. So fragment antigen binding because they had Ag binding activity. Fab are considered to be univalent, possessing one binding site each and being in every way identical to each other. The third fragment of molecular weight, 15,000, is called Fc fragment. Fragment crystallizable. It cannot bind antigen. At about the same time, Edelman discovered that when gamma globulin was extensively reduced by treatment with mercaptoethanol, the molecule fell apart into 4 chains, 2 identical light chains with a molecular weight of about 53,000 Dalton each and 2 others of about 22,000 Dalton each. The larger molecules were designated heavy chains and the smaller ones light chains. And on the basis of this results, the structure of immunoglobulin molecule was proposed. This model was subsequently shown to be essentially correct, and Porter and Edelman shared the Nobel Prize for the elucidation of antibody structure. Another researcher, Nisonov, used pepsin to digest the Ig. It generated a single 100,000 mwatt fragment composed of 2 Fab-like fragments and designated, unlike Fab fragments, FAB2 fragments was able to precipitate antigens. The Fc fragment was not covered from pepsin digestion because it had been digested into multiple fragments. Antigen binding and antigen binding. Here we could see a picture. Schematic diagram of a structure of immunoglobulin. Here we could see this. Okay, antigen binding portions. Fab Fc regions. Minor differences in the amino acid sequences of alpha and gamma heavy chains lead to further classification of the heavy chains into subclasses. In humans, there are 2 subclasses of heavy chains, alpha 1 and alpha 2, and 4 subclasses of gamma heavy chains, gamma 1, 2, 3, and 4. Then immunoglobulin domains. The structure of immunoglobulin molecule is determined by the primary, secondary, tertiary, and quaternary organizations of the protein. The primary structure, the amino acid sequence, accounts for the variable and constant regions of the heavy and light chains. The secondary structure is formed by folding of the extended polypeptide chain back and forth upon itself into an antiparallel beta pleated sheet. The chains are then folded into a tertiary structure of compact globular domains which are connected to neighboring domains by continuations of the polypeptide chain that lie outside the beta pleated sheets. Finally, the globular domains of adjacent heavy and light polypeptide chains interact in the quaternary structure, forming functional domains that enable the molecule to specifically bind antigen and at the same time perform a number of other biological effector functions. Both light and heavy chains are made up of repeating segments, each about 110 amino acid long and each containing one interchain disulfide bond. Both light and heavy chains are made up of repeating segments, each about 110 amino acids long and each containing one intra-chain disulfide bond. These repeating segments fold independently to form compact functional units called immunoglobulin Ig domains. The light chain consists of one variable and one constant domain. So, antigenic determinants on immunoglobulins. Since antibodies are glycoproteins, they can themselves function as potent immunogens to induce an antibody response. An antigenic determinant or epitope on Ig molecule fall into 3 major categories which are located in characteristic portions of the molecule. So, isotype and antibody class is determined by the constant region sequence of the heavy chain. The 5 human isotopes designated IgA, IgT, IgE, IgG, and IgM exhibit structural and functional differences. So, isotype determinants are constant region determinants that collectively define each heavy chain class and subclass within a species. Each isotype is encoded by a separate constant region gene and all members of a species carry the same constant region gene. Different species inherit different constant region genes and therefore express different isotopes. Therefore, when an antibody from one species is injected into another species, the isotope determinant will be recognized as foreign, inducing an antibody response to an isotypic determinants in the foreign antibody. IgG and IgA isotype differences. Then allotype. It is based on genetic differences among individuals. It depends on the existence of allelic forms of the same gene. These alleles encode minor amino acid differences called allotypic determinants that occur in same but not all members of a species. As a result of allotypy, a heavy or light chain constituent of any immunoglobulin can be presented in some members of a species and absent in others. The situation contrasts with that of immunoglobulin classes or subclasses which are present in all members of a species. IgG1, IgG2 allotypic differences we could see. Then idiotype. The unique amino acid sequence of a variable domain of a given antibody can function not only as an antigen binding site but also as a set of antigenic determinants. Antigen binding sites and antigen determinants. The idiotypic determinants are generated by the conformation of the heavy and light chain variable regions. Each individual antigenic determinant of the variable region is referred to as an idiotype. IgG, idiotype differences. So according to the clonal selection theory, all the immunoglobulin molecules on a single B cell have the same antigenic specificity. Explain why the presence of both IgM and IgD on the same B cell does not violate the unispecificity implied by clonal selection. So IgM and IgD differ in their constant region domains, whereas antigenic specificity is determined by the variable region domains. So molecules of IgM and IgD that have different D domains but identical VH and VL domains are found on a given B cell. Thus the cell is unispecific although it bears two isotypes. Then in different classes of immunoglobulin. In mammals, there are 5 classes of antibodies: IgA, D, E, G, and M, each with its own class of heavy chain alpha, beta, gamma, delta, and mu respectively. In addition, there are a number of subclasses IgG and IgA immunoglobulins. For example, there are 4 human IgG subclasses: IgG1, IgG2, IgG3, and IgG4, having this kind of gamma 1, gamma 2, gamma 3, and gamma 4 heavy chains respectively. Each class and subclass has characteristic properties of its own. Except for their variable regions, all the immunoglobulins within a class have about 90% homology in their amino acid sequences, but only 60% homology exists between classes IgG and IgA. So IgG is the most abundant Ig in serum, constituting about 80% of the total serum Ig. There are 4 human IgG subclasses distinguished by differences in gamma chain sequence and numbered according to their decreasing average serum concentrations: IgG1, IgG2, IgG3, and IgG4. Except for the IgG3 subclass, which has a rapid turnover with a half-life of 7 days. The half-life of IgG is approximately 23 days, which is the longest half-life of all immunoglobulin isotypes. The IgG isotype, except for subclass IgG2, is the only class of immunoglobulin that can pass through the placenta, enabling the mother to transfer her immunity to the fetus. Which one? IgG. Then IgM. IgM is the first class of antibody made by a developing B cell. It accounts of 5 to 10% of the total serum Ig. IgM is also found on the surface of mature B cells together with IgD, where it serves as an antigen-specific BCR. So monomeric IgM with a molecular weight of 180,000 is expressed as membrane-bound antibody on B cell. IgM is secreted by plasma cells as a pentameric molecule composed of 5 such units, each of which consists of 2 light and 2 heavy chains, all joined together by additional disulfide bonds between their Fc portions and by a polypeptide chain termed the J chain. The J chain is synthesized in B cell or plasma cell. The half-life of IgM molecule is approximately 5 days. So IgM is the first immunoglobulin class produced in a primary response to an antigen, and it is also the first IgB to be synthesized by the neonate. IgM is more efficient than IgG in an activating complement. So complement activation requires 2 Fc regions in close proximity, and the pentameric structure of a single molecule of IgM fulfills this requirement. Then IgA. IgA constitutes 10 to 15% of the total Ig in serum. It is the predominant Ig class in external sections such as breast milk, saliva, tears, and mucus of the bronchial genitourinary and digestive tracts. The IgA class of immunoglobulins contain two subclasses, IgA1 93% and IgA2 70%. Serum IgA has a half-life of 5.5 days. The IgA present in serum is predominantly monomeric. The IgA of external secretion is called secretory IgA. Consists of a dimer or tetramer with a J chain polypeptide and a polypeptide chain called secretory component. The daily production of secretory IgA is the maximum among other IgA. It is the major immunoglobulin found in the colostrum of milk in nursing mothers, and it may provide the neonate with a major source of protection against pathogens during the first few weeks after birth. Secondary component, J chain IgA dimer. Here you could see IgE also. It mediates the immediate hypersensitivity reactions that are responsible for the symptoms of hay fever, asthma, hives, and anaphylactic shock. IgE binds to Fc receptors on the membranes of blood basophils and tissue mast cells. So cross-linkage of receptor-bound IgE molecules by the antigen allergen induces degranulation of basophils and mast cells as a result of a variety of pharmacologically active mediators giving rise to allergic manifestations. IgE-mediated granulation is necessary for antiparasitic defense. And IgD, it constitutes 0.2% of the total Ig in serum. So IgD together with IgM is the major membrane-bound Ig expressed by mature B cells. It is thought to function in the activation of B cells by Ig. No biological effector function has been identified for IgD. IgG, which contains alpha heavy chains. So IgG, which contains gamma heavy chains, developed. IgG, which contains gamma heavy chains, developed much more recently during evolution than IgM, which contains mu heavy chains. Describe two advantages and two disadvantages that IgG has in comparison with IgM. So advantages of IgG compared with IgM are its ability to cross the placenta and protect the developing fetus. Its higher serum concentration, which results in IgG antibodies binding go and neutralizing more antigen molecule and being more effective in antigen clearance. So its smaller size, which enables IgG to diffuse more readily into intercellular fluids. The disadvantages of IgG compared with IgM are its lower capacity to agglutinate antigens and activate the complement system, both of which are due to the lower valency of IgG. An action of antibody, in addition to binding antigens, antibodies participate in broad range of effector functions that will result in removal of antigens and death of the pathogens. Because of these effector functions result from interactions between heavy chain constant regions and other serum proteins or cell membrane receptors. Not all classes of antibodies have the inner functional properties. Some major antibody-mediated effector functions are given below. Opsonization. Opsonization is the process by which microorganisms or other foreign particles are coated with antibody and or complement and thus prepared for recognition and ingestion by phagocytic cells. So opsonizing antibodies, IgG, bind to Fc receptors on the surface of macrophages and neutrophils. This binding provides the phagocyte a method to capture antigens. Then toxin viral neutralization. Some gram-positive and gram-negative bacteria produce extracellular toxins that contribute to their pathogenic effects. So immunity against such toxin depends on the production of specific antibodies that inactivate the toxins. This process is called toxin neutralization. Once neutralized, the toxin antibody complex is either unable to attach to receptor sites on host target cells and is unable to enter the cell, or it is ingested by macrophages. An antibody capable of neutralizing a toxin is called antitoxin. Similarly, antibody like IgG, IgM, and IgA can bind to some viruses during their extracellular phases and inactivate them. This antibody mediated viral inactivation is called viral neutralization. And viral neutralization prevents a viral infection due to the inability of virus to bind to and enter its target cell. And activation of complement. Complement is a set of plasma proteins that can be activated either by binding to certain pathogens or by binding to antibody. So complement activation is often described as a series of cascading enzymatic events leading to the generation of specific complement components that cause opsonization and phagocytosis to infectious agents as well as direct lysis of the invading organisms. IgM and most IgG subclasses can activate the complement system. Then immune complex formation. The interaction of antigen with antibodies may result in a variety of consequences, including precipitation and agglutination. Precipitation results from the interaction of a soluble antibody with a soluble antigen to form an insoluble complex. So antibodies that disaggregate soluble antigens are called precipitants. So the reactions of antibody with a multivalent antigen, that is particulate and insoluble particle, results in the cross-linking of various antigen particles by the antibodies. This cross-linking eventually results in the clumping or agglutination of an antigen particle by the antibodies. Then next topic we will be learning with the help of next video. Thank you for listening.